Team Brain Blast Junior Colloquia presentation. I'm Kelly Scourge, this is Mayumi Rezwan, Uti Mehta, and Alana Green, and we'll be your presenters tonight. I'd like to start out this presentation by saying a little something about our former mentor, Dr. B, who unfortunately passed away earlier this semester. Dr. Biquette, or Dr. B as we called him, was with us for the first year and a half of our research. At the beginning, we really struggled to narrow down our idea, and Dr. B, being an expert in metabolomics in the animal science department, really helped guide our project to what it is today. But the term mentor doesn't really cover the relationship that he had with our team. You can see in these two pictures over here, these were taken when our team first got together at the Ropes Challenge course at Epley Recreation Center. Dr. B had so much fun with us, and not only was he there to cheer us on, but he even attempted the course as well. He'll be greatly missed. He really got to know each one of us, and we owe him a lot of gratitude for getting our project to where it is today. So this is our current team. Our new mentor, Dr. Duffy, has been our expert for the course of our project so far, and we're really fortunate to have her as our mentor right now. These are our 12 team members. We come from a very diverse variety of majors, coming from anywhere from neurophysiology to engineering to business. Okay, so we're going to tell you a little bit about what we've done. First, we're going to talk to you about Alzheimer's disease and why it is such an important problem to study. Next, we're going to tell you about the specific questions that we attempt to answer and how we're going to answer those in our experimental setup. Last, we're going to tell you about our current accomplishments, future plans, and advice we have for upcoming teams. So with that, I'm going to hand it over to Mayumi, who will talk about the background information.
Thanks, Callis. So as a team, it was really important to us to pick a research topic that has a large impact on society, and Alzheimer's disease is just that. More than 5 million people live with Alzheimer's disease, and not only is this really devastating for those inflicted with the disease, but it can have a huge financial and emotional toll on caregivers as well. It also has a large impact on the national economy, with costs going over $200 billion. But most importantly, approximately 500,000 people die every year due to Alzheimer's, making it the 6th leading cause of death in the United States. Moreover, it is the only cause of death in the top 10 that does not have a cure and cannot be prevented. So as we move on with our presentation with more science and research-based information, we ask you to keep these figures in mind, just as a reminder of how real this disease is for so many people and why we think it's so important to research it. So what exactly is Alzheimer's disease? On the cellular level, it can be traced down to 3 main things. Amyloid beta plaques can form when the amyloid beta protein is broken down incorrectly. So these sticky plaques can form in the synapses between the neurons and can block cell-to-cell communication. The protein tau is responsible for maintaining the structure of the cell. However, when it's hyperphosphorylated, it can prevent this protein from doing its function and can actually lead to the breakdown of the transportation system in the cell. Finally, neuron loss and cell death is a large part of Alzheimer's. As you can see from the figure, an Alzheimer's brain will be much more shrunken than that of a healthy brain. So what all of that means is that Alzheimer's occurs when there is a breakdown of the function in the brain. It is the most common form of dementia and causes problems in memory, thinking, and behavior. It is neurodegenerative, which means that the brain is breaking down slowly and the condition worsens over time. So the current research is focusing on what exactly causes this breakdown of the brain. A lot of this research is pointing towards Alzheimer's being considered type 3 diabetes, and that is because the problems of glucose uptake in the brain in Alzheimer's is very similar to the glucose uptake problems in the body in diabetes. So that's why a lot of studies are now focusing on using diabetes treatments, such as insulin, on the Alzheimer's brain. Another example of this is the use of thiazolidinediones, or TZDs, which is a class of diabetes drug. So in a healthy body, insulin binds to insulin receptors, and that basically signals the cell to take in glucose. However, in diabetes, these insulin receptors are desensitized. What TZDs do is that they activate PPAR gamma, which is part of the insulin signaling pathway, and that signals the cell to take up more glucose. The most common form of TZDs that are used for Alzheimer's research is pioglitazone, and that's just because that is the most healthy option. So as I mentioned a bit before, there's a lot of research focusing on administering insulin straight to the brain for Alzheimer's research, and that is mainly done through the intranasal method. And that is because this method bypasses the blood-brain barrier, which is just what it sounds like. It is a barrier that keeps things from entering or leaving the brain to the body. However, if something might not be able to bypass this barrier naturally, if it is administered through the nose and straight to the brain, it avoids this barrier altogether. So as you can see, there's a lot of research being done. However, there's still some gaps. For example, while insulin is very commonly administered to the brain, other drugs such as TZDs have not yet been tried to be delivered intranasally. TZDs, so far in research, has only been administered orally into the body. However, this can cause a lot of problems because TZD molecules are too large to bypass the blood-brain barrier. So if they are administered to the body, they do not have the wanted effects in the brain and can actually have unwanted effects in the body. So these are some unanswered questions that we hope to explore in our study, which I will allow Kuniti to explain in further detail.
Thanks, Mayumi. So I'll be going over our research questions, hypothesis, our pilot study, and our main study. So first, our research questions. There are three main questions that we hope to address with our research. First, does intranasal pioglitazone increase glucose metabolism in the brain? Essentially, we want to look at whether pioglitazone increases glucose uptake in the brain without increasing peripheral effects, which are effects on the rest of the body. Then, we want to look at whether the combination of intranasal insulin and pioglitazone have an additive effect on glucose metabolism in the brain in an Alzheimer's disease mouse model. So basically, we want to see if the combination of insulin and pioglitazone increases glucose uptake more than if we were to just give pioglitazone. And finally, following a prolonged period of treatment with insulin and pioglitazone, we want to see if there are any lasting neurochemical changes and a reduction in Alzheimer's disease pathology if we give the pioglitazone for a long period of time. So our study is split up into a pilot study and a main study. And our pilot study will be using normal C57 mice that do not demonstrate Alzheimer's disease pathology. Our pilot study has two phases. The first phase is pilot 1, and with that, we want to determine the optimal dose of pioglitazone to give to the mice per dose. So with this, we have 4 groups. Each group of mice will get a different dose of pioglitazone, and they will all be sacrificed after 36 hours. Sacrifice is the official term for kill. After we sacrifice these mice, we will measure glucose levels in the brain to see which dose gives the highest level of glucose uptake. Then, we will move on to pilot 2, which will determine how long one dose of pioglitazone lasts in the brain. So for this, we have 3 groups, and we will give them a dose of either saline, insulin, or insulin and pioglitazone, and sacrifice them after either 1 day, 2 days, or 4 days. Because we want to minimize the amount of administrations of pioglitazone we give to the mice, we will determine our frequency of dosage based on this study, because the highest number of days that we see the longest lasting of pioglitazone, that's the dosage that we'll go with, the frequency of dosage. So now our main study has 3 phases. Our first phase is basically going to be examining how much glucose uptake increases in an Alzheimer's disease mouse model. So it's essentially parallel to pilot 1, but we will have 3 experimental groups, 1 receiving saline, 1 receiving insulin, and 1 receiving insulin and pioglitazone. And we will sacrifice all 3 groups after 36 hours to see how glucose uptake works in all 3 of the groups. Then for our second phase, we're going to see how long-term administration of pioglitazone decreases Alzheimer's disease pathology. So we're going to dose the mice, not just once, but over 21 days with the frequency that we established from pilot 1. We'll sacrifice all the mice after 36 hours and measure Alzheimer's disease pathology. And finally, for phase 3, we want to examine how long-term administration of pioglitazone has a lasting effect even after treatment. So the only difference between phase 2 and phase 3 is that rather than sacrificing the mice after 36 hours, we will sacrifice them after 2 weeks to see how long the effects of pioglitazone remain. So to summarize, we are looking for If pioglitazone increases glucose uptake in an Alzheimer's disease mouse model, whether long-term administration of pioglitazone decreases Alzheimer's disease pathology, and whether long-term administration of pioglitazone has lasting effects even after treatment. Now I'll hand it off to Alana to talk about our research progress and methods of analysis.
Thank you. So as Anissa just said, I'm going to discuss with you guys how we're going to measure the reduction in Alzheimer's pathology as well as the change in glucose metabolism, and then I'm going to update you on what we have discovered from completing our pilot 1. So first, we're going to discuss measuring Alzheimer's pathology. So there are a few different things we're going to be measuring, as Mayumi had discussed earlier. First, one of the main markers of Alzheimer's disease is the presence of amyloid beta plaques and the hyperphosphorylated tau protein. So the way we're going to measure the abundance of that in the brain is that we're going to use antibodies that are very specific for these proteins. And the higher the number of antibodies that bind to the proteins means that the pathology is more advanced, so the Alzheimer's disease has gotten worse. We're also going to be measuring the amount of cell death or cell degeneration as well as the amount of cell regeneration. In more advanced Alzheimer's, you would expect to see more cell death and less cell regeneration. So similar to how we're going to be measuring the amyloid beta plaques and the tau proteins, we are going to be using specific antibodies that bind to proteins in cells that are excreted upon cell death and cell regeneration. So here I'm going to talk to you about how we're measuring glucose metabolism using a method known as flexomics, which is very highly inspired by our mentor, Dr. B. This is his specialty. So the way to do this is glucose turns into energy essentially by going through metabolism, and there are many intermediary molecules known as metabolites. And some of these metabolites are listed in this cycle here, which is called the citric acid cycle. So we want a way to measure the rate of those molecules that are metabolized in a certain amount of time, looking for a higher rate, hopefully if they have more pioglitazone administered. So the way we're going to track that is that we are going to use a special glucose where all the carbons have 13 neutrons rather than 12. So we can actually use a method called gas spectrometry, which can detect different types of molecules and their relative abundances, and they do that by detecting the weight of the molecule. So if we expect to see more carbon-13 labeled metabolites if the glucose metabolism rate is higher. So right now I'm going to walk you through what we've done so far. We have completed our pilot 1, where, going back to what Unuti said earlier, we are trying to find the optimal dose of pioglitazone. Shortly before sacrificing the mice, we gave them a carbon-13 labeled glucose so that could go through their bodies so we could use the flexomics method, which I just described. We ultimately harvested blood and brain samples and derivatized those samples so we could run them in our flexomics analysis. So here are the preliminary results of our findings. We've not finished looking over the results, but we have enough data in order to explain the trends in the data to you all. So first of all, flexomics analysis has never been done with brain tissue before, so the fact that we even got this far with our analysis is a good sign. If you look at the graph, you can see that we are increasing the amount of pioglitazone and measuring the percent of carbon-13 in the metabolites that we measured. As you can see, the blood metabolites did not have an increase in the amount of carbon-13 metabolites, which shows that there were no peripheral effects from our intranasal administration, which is a really good sign because that's what we were expecting by giving the intranasal dosing. The pioglitazone did not go back from the brain into the body. If you look at the brain metabolites and the amount of carbon-13, you can see that there is an increase with the amount of pioglitazone we gave the mice. So in an effort to figure out the exact dose we wanted to give them, we had to compromise between the best glucose metabolism and also avoiding any negative effects we saw. And we saw that the 60 microgram pioglitazone actually caused some mice to show aggressive behavior, and in a long-term study, we didn't want mice to be aggressive, so the team determined to go with the 30 microgram pioglitazone. And I'm going to hand it over to Kellis to finish things up.
Thank you. I'm going to briefly talk to you guys about our timeline and then a little bit of advice we have for the upcoming teams. So over the next couple semesters, this is our timeline. In the spring, we will have hopefully completed all of our testing, and then we will begin analysis. In the spring, we will also present at undergraduate research day, and then potentially at other conferences in the future. In our senior year, we will focus on writing our thesis and then presenting at Senior Thesis Conference. So here's the advice that we have for upcoming teams. First of all, building relationships within your team is so important, and more important to that is trust. This will go a long way in the 3 years that you'll be working together. Next, you have to really prepare for your unexpected challenges. There are some things that you can prepare for and some that you can't. But for those that you can't, you just have to work around it and do your best as a team. Next, we recommend that you use the resources, the many resources that you have on campus. Not only do you have librarians and experts, but you also have the Gemstone staff. I know as a team, we've personally gone through several team building workshops with Dr. Skendel, and that's really paid off in the long run. Lastly, it's really important to harness the individual differences and make those work for you. It's important that you're open to change and, more importantly, compromise. We've undergone some challenges as a team. Some of our current challenges include the age of our mice. Because there's such a small window in the peak pathology of our mice, this has taken a lot of planning as a team. Also, we're waiting for our current UCOOC amendment to go through the approval stage. Lastly, the learning never stops in Gemstone. Just because you've finished your proposal, you still need to constantly update your information and always keep learning. Okay, so we'd like to extend a thank you to our mentor, Dr. Duffy, Dr. Cole, and Dr. Skendel, and the Gemstone staff, our UCOOC PI, Dr. Kastangwe, the Animal Sciences Department, and our librarian, Madalena Changuoka. Thank you. I'd like to open the floor up to questions. Yes, in the back.
So it said that you were using a model for Alzheimer's disease in mice. What exactly is like the type of mouse that you're using, and how is that model, like, how is that similar to Alzheimer's disease?
Okay, so you're asking what is the mouse model we're using and how is that related to Alzheimer's disease?
So we're actually using a colony of Alzheimer's mice. They're triple transgenic, which means that they have 3 genes modified to make sure that they have familial Alzheimer's. And since part of Alzheimer's is genetic, it's passed down from mouse to mouse. So the entire colony has Alzheimer's, and many researchers have proven that the pathology in the brain of the mice is very similar to that of Alzheimer's in humans.
Why do you believe the 60 microgram dose ended up causing aggression in the mice?
That's just what we observed in our study. Well, because we go out to check the mice every day.
So we're not exactly sure why they would show aggressive behavior, but some of us are postulating that that might have increased glucose metabolism to a point where they were so high energy that they were acting aggressive because they had so much energy.
What is the reason for the faulty breakdown of amyloid beta?
And what is the reason for the faulty breakdown of amyloid beta?
That's actually kind of what's unknown about Alzheimer's disease. We don't know why the amyloid beta plaques are conglomerating and causing those blockages. And with our study, if we do find that there is decreased Alzheimer's pathology, we'll be able to postulate that downstream of PPAR gamma in the insulin signaling pathway may be a cause for that. So that is one of the things we have to contribute.
Yes?
If you were going to test a standard colony of mice, one that hasn't been bred to show signs of Alzheimer's, would you expect a change in their actions or a change in their brains if they underwent the same treatment? Or is this purely for ones that would exhibit Alzheimer's?
So as our pilot study shows, pioglitazone does increase glucose metabolism in the brain based on our preliminary results like Alana talked about. So it's not only in Alzheimer's disease mice, but we hope that the increase of glucose uptake in the mice with pathology, that will improve their condition.
Yes, in the way back?
I guess mine has kind of 2 parts. The first part is, how was the labeled glucose administered into the system? Was it fed to them or was it injected with the drug?
It was injected intraperitoneally into the mice.
have you considered the fact that glucose also needs to be transferred through the blood-brain barrier, and that the fact that you're directly administering it, of course, is going to show up in all of the metabolites within the brain because that's how it's going to be metabolized?
Yeah, so we administer it through the abdomen, and glucose can pass the blood-brain barrier because it is smaller enough and has all those properties. So the fact that the peripheral blood levels of the carbon-13 labeled metabolites showed consistent, because there is going to be glucose metabolism everywhere in the body with that labeled glucose, the fact that that was consistent no matter what the pioglitazone dose was showed that that it increased in the brain and only in the brain. Does that answer your question?
So, how do you determine that the 36-hour period from the dose to the cytoplasmic analysis was the optimal period?
That was mostly what was consistent in the literature. Yeah, we've done so much research, we've looked at probably hundreds of articles, and so we took the most consistent time. And one more question? Yes.
You said you guys are going to measure the amount of drug after 2 weeks to see its lifespan in the brain. Have you guys given thought to maybe doing different time points to see like a release profile in the brain?
So his question was about the different times that we're going to be testing.
Okay, so originally our plan had been to test different time points after treatment of about 1 week, 2 weeks, and even 8 weeks after. And like we talked about before, there's unexpected challenges that you're going to face as a Gemstone team. So that was one of the things we had to recently change in our methodology. So now we're just going to look at 2 weeks, and it's because the challenges we're facing with the mice's age are not going to allow us to be able to test those different time points anymore.
Thank you.